Thank you. Thank you for having me, guys. I'm excited to give this talk as well. There's been some very good implementation of the Aurox system before, even shared by Michael. And I'm hoping to show my side of things and what I'm using it for in terms of looking at cell and vesicle biology. So just to give a brief background on the need for cryofluorescence imaging, specifically for me as well in my applications in biosciences. So it's known that cryogenic fluorescence is important to in a way that it improves the fluorophore lifetime. Is also good for high resolution imaging. And cryopreservation itself gives the best quality of biomaterial preservation, gives us a frozen hydrated state, particularly good for looking at 3D images of cells and tissues. And finally, for my platform, loads of native ultrastructural and compositional analysis requires cryogenic imaging, and encompassing and including a fluorescence module is really important because, as we know, it gives a multi-contrast ability where we can study multiple molecules at the same time, and we can correlate it with between different imaging modules to bring more information about the biology that we want to learn and see. So Michael had given a brief overview about his setup, and this is a kind of different setup that I made in Nottingham. So with the help of the Schools of Engineering and of course Aurox team as well. So first thing I wanted to do was to have a cryogenic stage that can be imaged at fluorescence on fluorescence value. So this is an ACoM2021 upright microscope, and the first thing I did was to install the Lincoln cryo light stage. So inside the stage, we would have the grid boxes and sample transfer abilities that Michael had showed before, and it also has a manual XY navigation system. So initially, when I was clicking images, I found that there was a lot of out of focus light, and being in cryo, you get a little bit of a background that is hard to get rid of. And one way to do what I was doing was to manually move the focus up and then do a semi-deconvolution to improve the clarity of the image. However, this was taking a lot of time, and it was also highly error prone in terms of human manipulation. So we had a chance to attach the clarity laser-free confocal system on top. The only thing we had to do in terms of hardware is to make a custom adapter, which helped fit it perfectly. So after that, we had a astonishing difference in how our image quality was, specifically looking at subcellular events and information. The next step for us was to make the system into a 3D platform. So we attached a MFC focus knob attached Z controller. So this is completely motorized, and now we can do optical sectioning at the nanoscale or at the submicron scale. Now, currently, we're also fitting in an automated Lincoln stage that will allow automated tile scanning and capturing a correlative map of our whole image sequence, and then we can use that to map between instruments such as electron microscope. Now, to make all of this happen, Philip and Cesare really helped us out and made sure that the software was talking to each other and everything is combined into one platform so that it's easy and user-friendly and robust. So this is an example of how our images look like. In this, on the left here, we're looking at nuclear invaginations in cancer cells. So this is a MCF7 breast cancer cell labeled with an endoplasmic reticulum dye, and basically allows us to look at nanoscale nuclear pore invaginations. And we can see how we can look at it in 3D. And for my interest, I look at vesicles as well, so I approached on that on another side. On the other hand, we are also looking at tissue biology. So these are high pressure frozen vascular tissues. On the top here, you are looking at retinal vessels labeled in DAPI blue. And in the bottom, you're looking at kidney tissues, which is also taken by the cryo-confocal microscope. You can look at the tubules, you can look at the glomeruli, all in cryopreserved format. So because I work in correlative microscopy, I develop correlative platforms as well. It's a very straightforward method. In order to identify our cellular as tissue and as well as subcellular regions of interest between light microscope and electron microscopy, you need to have marks which can be identifiable between these instruments. So for cells and vesicles, for example, what I do is to culture my cell samples on grids, or put my nanoparticles on these correlative grids. You can either purchase them without a film, and then you can manually add the film. For example, I've added a pioliform film, or we can purchase it directly as a carbon-coated film on which the cells can grow. And I usually treat with a substrate, for example, matrigel or polylysine, so that the cells are happily attached and growing. And basically, you can figure out between these bright field and fluorescent microscopes, you can look at the nuclear area based on these correlative marks, such as S and dots over here. When it comes to tissues, you have to high pressure freeze them. For cells and vesicles, you plunge freeze them, but for tissues, they're thicker, so you high pressure freeze them. And we do them in either gold or aluminum planchettes. And these planchettes don't usually come up with a correlative mark. And to make sure I'm able to adapt it to the current system, I custom make my marks. So basically what you're looking at here is using a scalpel or a metal pen, I just make these marks on the corners of these planches before sandwiching them with the sample, which are also fluorescent. And then, as you can see, we'd be able to relocate these areas under electron or in fluorescence, as well as in ion imaging platforms. So what am I using the confocal system for? So most of or majority of the correlative platforms look at light and electron imaging to look at structural properties of in biology and materials. And I'm also interested in looking at compositional analysis, and particularly compositional analysis, correlative analysis at the nanoscale. So this image here shows how I've used the cryo-confocal setup to identify regions, cellular and subcellular regions of interest. Here in blue, you're looking at DAPI. And then I was able to move this sample into a mass spectrometry instrument. So this is a newly established hybrid SIMS instrument. And basically, you're able to get thousands of chemistries out of it in 3D nanoscale, because this instrument can go up to below 50 nanometers in around 50 nanometers in spatial and 2 nanometers in depth. And to look at gene analysis, such as transcriptomics, I developed another method, and cryo-confocal was central to this platform here. So basically what I did was to relocate subcellular regions of interest using fluorescence in cryo-focused ion beam microscope. And precisely here, shown in sub-micron 3D scale, I was able to isolate subcellular regions and then transfer them to a PCR tube, and then get nanoscale and then get microscale transcriptomic information. Both of these instruments can go up to sub-nanoscale, so it's just a matter of time when we can understand the 3D molecular arrangements of genes within cellular compartments. So to make all of this happen, I also engineered a custom stage. So basically, you can clearly see that it can hold the Lincoln grid holder. It can also hold these planchettes. It can hold multiple samples, and essentially it has improved our throughput in correlative platform for, up to 8-fold, but also protecting the sample from damage and contamination. I just put one image here to show my interests in vesicle analysis. So I'm a big fan of extracellular vesicles. So I've used the cryo-confocal first in non-cryo mode, actually, to identify single vesicles and correlate their structure. Basically, with a simple single image, you'd be able to get the purity, heterogeneity, and also the labeling efficiency of vesicles in high quality. We have moved to do this in cryo, but we are still improving the platform to get rid of background. So this slide is sort of a summary slide showing how essential and central the cryogenic adaptable microscope has become. So as an alternative to integrated systems that you may find in other places as well. So efficiently, the sample gets repetitively transferred between these multiple electron and ion imaging instruments and the cryo-confocal to to not only locate the samples, but also study the functional regions of interest that we are looking at in terms of cell and tissue biology. So we were able to put this together as a Wiley article, which got published recently. And I'd like to now acknowledge the lab I worked in to develop this. So it was in Kenton's lab, and he has an interesting way of making abbreviations. So for example, he calls it the EVIL lab, endothelial and vascular imaging lab, and all my colleagues who worked with me. Also, a big shout out to the Nanoscale and Microscale Research Center, particularly Chris, Julie, and David Skirb, and also the School of Life Sciences at Nottingham, particularly Robert Marcus, who's helped me a lot with imaging and still continues to help me out, and also Tim, who leads the group. And also a big thanks to the Aurox team to make all of this happen in a short amount of time. So thank you for listening, and I'm happy to answer questions.
Thank you so much, Zubair. It's been an absolute pleasure working with you the last few years on getting this system up and running. I remember when Kenton first got his funding to buy an Aurox system, and we've moved it to and from various microscopes a lot, haven't we, between us on a trolley in the rain. And I'm really excited to see what you guys can achieve with the motorized stage. I think that's going to really open up your multiple regions of interest, your tile scans, and things like that. So I'm just seeing if there are any questions in the chat window. We're just waiting for Stephanie to join because we're obviously running a little bit early. So what are you planning to do in the future, Zubair? I know that you've changed roles a little bit, but I hope you're going to carry on with this cryofluorescence research as well. So where do you see this system and this technique moving forward?
It will, I think it will become a standard and established suite for all types of correlative work that includes fluorescent identification of molecules. But of interest to me, I'm looking at single vesicle characterization. I'm trying to push the correlative platform to look at subcellular organelles and trafficking of these small molecules. So it will be essential for the cryo-confocal platform to be fully automated, because we'll be looking at different regions within cell at probably the highest magnification we can get. And then we'd be it will not be a one-step go. So once we identify a region of interest, I think we'd be processing it in the cryo-FIB-SIM and picking out our nanoscale lamellas out, and then we'd be re-imaging them to make sure that we've got the right thing. So a lot of sample protection, sample transfer, especially in a non-integrated system, those sorts of things I'd like to develop so that things become really routine. Particularly in the 3D nanoscale transcriptomics, I'm just excited to see how flexible the platform is going to be, what are the problems we are going to come across, the hardware and software, and also it's a cost-effective platform. So to see like how it compares in and sets itself up as a center to do such analysis. So yeah, I did move across to do more biology, but my interest in imaging still applies to, and I'm still working with all of my colleagues and collaborators. It's not like moving on, it's still a progress. Yeah,
that's good to hear. We've got a question in the chat folder from Mariana Cavallo. She says, from the full process, what would you say is the hardest part to work with from your full pipeline? What's the hardest bit?
I mean, I only have like 2 years of experience, but I would say when I started from scratch, just handling grids and handling these precious samples under liquid nitrogen, making sure I don't lose them was really hard in the beginning. But then you get better at it. But alternatively, also develop these stages, and that can actually reduce the amount of times you meddle with these things. So that improved the throughput and efficiency a long way, so that not just me but others can also come across and use it immediately. For example, the linkum stage holder really helps not destroying the sample again by touching the grids, and the platform can be easily relocated in other areas. So that was okay. And in terms of software, I think Philippines has really just finished it up for me. Otherwise, I would have struggled a lot.
Yeah, well, we were integrating lots of hardware together that doesn't normally talk to each other, so it took a little bit of an effort at the beginning, but it seems to be all working now. So that's really, really good. And these talk me through transferring this subcellular region of interest. I've not seen the instrumentation that you use to do that. It looks very, very complicated to pick out this tiny region of interest and transfer it.
So usually the cryo-focused ion beam microscope is, as far as I know, from a structural point of view, they use it to isolate a small thin lamella or biopsy, if you want to call it, and they thin it to look at under the transmission electron microscope. And my idea was it's cryopreserved, so the biochemistry should be retained as well, and you should be able to analyze it. So it's actually easier to do what I'm doing than the actual structural analysis. So because I'm just roughly taking the area of interest where I found a fluorescent biomolecule, and then using a micro needle, I just go and touch it. It attaches itself, or you can use water vapor, and then put it into a plastic sheet on the corner. And basically, it looks complicated, but it's very crude and rough because it's like a prototype demonstration. I just lift it out and put it into a PCR tube quickly. So,
okay. It looks more complicated than it is. Then
the core is that you can analyze RNA straight away from such a native state of biology, and you can do multiple sections in the future and have like a transcriptome, but not just in 2D, but in 3D nanoscale, and you can correlate that with the Aurox's 3D visualization of your sample. And I think that's very exciting and very new, but I don't know how soon I can demonstrate to that extent.
Sure. Well, maybe next year you can present us with some more results. That would be really cool.